Right. Well, thank you everyone for joining today. What I will do in my part of the presentation is briefly give you the background of metagenomics and kind of following some of the resources that you will also find in the introductory materials on the Learn portal for anyone who would like to kind of get familiar with these concepts before we start the program. I want to kind of start from the very beginning and talk a little bit about, why the microbiome and how exactly can we study the microbiome. So since the discovery of the microscope, we've been learning about microorganisms that surround us and even live inside us. And with the increasing resolution, so here we have a chart of the different types of small things that you can see in the microscope. You can see that the optical microscopy resolution allows us to see kind of all the way to bacteria. And because of that, this kind of interest in the way that the microorganisms live together has evolved as the resolution allowed us to see smaller and smaller things. So what you can see here on the bottom is a figure. This is an image from a microscope, a super high resolution microscope, where you can see E. coli and some phages. So these are viruses that are sitting on the E. coli bacteria. So what has become clear with the increasing resolution is that not only that there are trillions of microorganisms around us, they also live in communities and they interact with each other, not just with their hosts or the environment. So because of that, early on, there has been an acknowledgement of the role that the microbes play in human health and well-being. And early on, you can see here in the mid-1600s, there was the first observation of microscopic organism. And by the 1800s, there has been an association made between disease and microorganisms. But today, in the last couple of decades, there has been a tremendous increase in research concerning the role of the microbiome in a variety of different conditions that affect the environment, but also affect us as people, including different aspects of human health and development of the healthy microbiome on the skin, in the gut, and in other organs. And what we will talk about today are several examples, just to kind of understand what are some of the considerations, how is the microbiome studied, what are some technologies that have been used to do that, how these technologies are evolving, and what we will cover in the program to be able to handle such data practically and kind of understand the insights that we can gain from such data. So one important factor about the microbiome is the stability. So essentially, these communities of bacteria, specifically we'll talk more about the bacteria rather than just the whole microbiome, we can kind of all relate to the stability of these communities when we think about such microbiome communities like our skin. So for example, microbiome on the skin will create a biofilm, and this biofilm can actually communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confers benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. And quorum sensing activities are also involved in the virulence and pathogenic potential of bacteria. And so they're an important factor in understanding and controlling bacteria and bacterial infections, as they enable microorganisms and biofilms to become more tolerant of host defenses and antimicrobial agents. And inside each community, there's going to be a balance of bacteria, fungi, and viruses that control species abundance in skin. So if you touch your own skin, you will feel that there is more than just one type of feeling on your skin, right? Some parts of your skin might be dry, some of them might be oily, some of them might feel smooth or might have bumps on them. So all of this means that there's this whole microbiome interacting with the host. And one question that we might have is, well, what factors are actually determining some of the qualities of this microbiome? How are these distinct communities different from each other? Because we know that all of our skins are probably very different from each other, but also how do the things that we can control affect these communities? And so to answer some of these questions, there has been a tremendous interest in understanding, characterization, and investigation of the factors that really control the microbial communities in many different organs. So you can see here on the screen some of those defined microbiomes, like the microbiome of the skin, the respiratory system, the gut, the intestines, the stomach, the urogenital tract. So all of these, since 2010, I believe, there have been hundreds of thousands of papers related to the microbiome and major projects that have been developed. So some of the major projects that you can see right here, in 2010, we have the Human Microbiome Project that we will talk a lot about in this program, but also throughout the years, there have been distinct projects focused on the gut, skin, lung, and even brain microbiomes. So many of these projects, like the Human Microbiome Project, seek to characterize the healthy microbiome because there's a lot of variation, as we already discussed and we'll talk about in a few slides, related to the age, to the location, to the diet and lifestyle, as well as the environment of the healthy microbiome. But then also an additional set of projects have been designed to address some of the questions around disease. So you can see here cancer prevention study for microbiome in cancer. You can see the Alzheimer's gut microbiome project, the nurses health study, and others. And so really we can see here a variety of disciplines that will be affected by the microbiome, from healthy nutrition and lifestyle all the way to a variety of precision medicine applications. And even you can see here on the bottom, space travel, right? So it appears to also play a role in space travel and the health of astronauts. And so many different areas of application for metagenomics and for microbiome. So because of all of these projects, we know today that the human microbial communities contribute to many critical and essential metabolic, physiological, and immunological functions. And they include differentiation and maturation of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosal barrier function, development and regulation of the immune system, prevention of invasion and growth of disease-promoting microorganisms, and several others. And so to study these conditions and this variability, there are several different types of omics technologies to study the microbiome. And these could be distinguished into sequencing-based or mass spectrometry-based. So for sequencing, we can look at the DNA-based approaches. So essentially, we can sequence specific genes or try to assemble whole genomes of bacteria or whatever species of these microorganisms. We can also look at RNA-based. So we can try to infer function. What are some of the genes that are being expressed? And what do they mean? What is their function? There are also ways to study the microbiome using mass spectrometry, where we can look at proteins and metabolites that they produce. But today, in this session, we'll talk a little bit more in detail about metagenomic sequencing. And so metagenomic sequencing Metagenomics in general could be characterized as both a field and a specific technique to study the microbiome. But it is different from many other types of omics. So essentially, many types of omics are focused on characterizing a single system, a single individual. For example, when we speak about the genomic studies, we're trying to understand the genome of an individual. Metagenomics, in that sense, is different because it looks at a community. So specifically, when we speak about a community, we think of the microbiome as almost like an organ that has a specific function. And one of the ways that we want to understand the function of this organ is by capturing the diversity of microorganism within that microbiome or microbiota, the community that it forms. And when we do that, it is different from a lab-based technique where essentially a single microorganism is isolated and is grown in a different environment than where it actually typically lives. And so because only 1 to 2% of bacteria can be cultured in the lab, metagenomics allows us to study how these microorganisms coexist together with others and respond together to different environmental changes or produce some functional effects. So the difference between these two, we can see here for example, when we want to study the gut microbiome, one of the ways that we can use sequencing to understand this microbial community is by trying to annotate the different taxonomic variation found within the gut. And sometimes we can also combine that with an understanding of a particular individual representative of that community to try to understand what is that specific individual bacteria, for example, how is it different from others, right? So we'll talk a little bit about how these techniques could be applied to different types of conditions in just a moment, but let's try to see how this type of information could be generated and what are some of the techniques to then study the data that is being generated from sequencing. So two kind of approaches that I want to discuss here. The first one is sequence-based, when we are able to sequence all of the DNA or the RNA that is present in a sample, like next generation sequencing. So this could be shotgun metagenomics, 16S amplicon metagenomics, or metatranscriptomics. And then sometimes we also have these approaches with qPCR when we know that we're looking for specific types of bacteria, and we could design primers to actually find whether there is an abundance of a particular type of a bacteria or combinations of bacteria. So if you think about the next few slides, you'll see how both of these could probably be applied to answer similar questions, but we'll more speak about next generation sequencing because of the richness of information that it can provide us with, and how we can apply qPCR, for example, to validate the findings that we will see in next generation sequencing. So really, shotgun metagenomics or metatranscriptomics differs from 16S amplicon metagenomics in the way that we're able to capture the specific view of the composition of the microbial community using 16S amplicon metagenomics, or essentially thinking about the 16S ribosomal RNA as a marker gene that helps us understand taxonomy. And then shotgun metagenomics or metatranscriptomics is the analysis of all of the genes within an ecosystem. So the difference is, are we sequencing all of the genes or all of the genome, or are we looking at conserved marker genes or gene regions? And some of the challenges that are associated with both of these approaches. So first of all, in trying to sequence all of the data that is present in the DNA that we extract from this microbial community, obviously assembly is going to be one of the major challenges. How do you take these short reads and try to piece them together to extend them into an assembled genome, or at least a part of the genome that then we can search for specific genes and try to annotate them. So even with 16S amplicon sequencing, we have this challenge of annotation as well, because essentially not all of the bacterial genomes are known, but because we have databases that are constantly being updated, because of these major studies, it is possible to define the taxonomy or the diversity and the presence or absence of specific types of bacteria in a given sample. So let's kind of talk about 16S metagenomic sequencing and try to understand the specific applications of 16S metagenomic sequencing, which I think are very exciting in the context of human health and well-being, different types of disease applications, and even behavior, as we will see. So 16S amplicon sequencing is a technique that is widely used, one of the most kind of widely used techniques. And this sequencing focuses on the 16S ribosomal RNA gene. So first, that gene is amplified with PCR amplification, and that gene contains hypervariable regions that allow us to study taxonomy of a particular community. So one of the challenges with the actual sequencing is first to organize the sequences that we get into what we call operational taxonomic units. And these are units that resemble each other with some specific markers. And then we can use those to annotate and try to understand what do they match in a database. And so there are several different databases like the Silva, Green Genes, there's the NCBI repository of all of the bacterial genomes, and as well as RDP. So what do these databases essentially provide us with? So these databases can help us understand taxonomy, or if we think about the hierarchy of biological classification, how we can study the more or less detailed view of the different types of organisms. So the taxonomy is just a way to organize the different types of organisms into some kind of a structure so we can see what is more related or less related. So starting from the bottom here, kingdom, phylum, class, order, family, genus, and all the way to species. Now, as you will see, not all of these are going to be available for all of the individual microbial organisms that we will have. And so what we can try to assess are the differences at maybe specific levels, like, for example, class, phylum, or order, or sometimes dig deeper into the species level of classification. So to really understand how diverse the microbiome is and how many organisms we're dealing with, let's actually look at the Silva database, and we'll see there how many different organisms you are going to be finding there. So I'm going to paste the link in the chat box, and I will actually address your questions here in a few minutes. So please go with me to this link. Okay, so this is the database, the Silva database. Here you will find the different levels So right here, you can see those levels: domain, phylum, family, major clade, class, genus, kingdom, and order. And so you can see here, bacteria, right? That's the kingdom. And then it goes all the way down to family right here. So what you can do here is you can search by sequences, by accession number, or by full name. And here you will be able to find by taxon name, or you can also browse here specific types of bacteria. So as we go through, I will give a couple of examples of bacteria that you will be able to find here and search for it right here in the taxon name, and then you'll be able to find it. Okay. Beyond the bacteria, right, what we need to remember is that a microbiome contains also viruses as well as other microorganisms. So because we have this interaction between bacteria, the community that it forms, and other microorganisms, right, we won't talk about it as much today, but you have to remember that the same types of databases exist for other microorganisms that you might expect to find in the microbiome. So again, viruses, for example, as well as you can see here, the virome, right? So in the same way that we can call the microbiome all of the microorganisms, we can also look at specific types of microorganisms. So for example, for viruses as well, called the virome. So let's think about how these different projects allow us now to study the microbiome, to understand, first of all, healthy microbiome and healthy variation, right? So we'll look at this left side of the screen, the Human Microbiome Project. You can see some examples here: American Gut Project, British Gut Project, etc. Okay, let me take a look at some of the questions. Okay, so are there any specific databases related to cancer, microbiome and cancer? So there are some studies that have shown specific variation in the microbiome that can lead to more successful treatment, for example, with immunotherapy, right? We'll talk about this in a little bit, but essentially, one thing that we need to understand is that we need to have a healthy baseline, right? When you think of any pathology, any condition, you're comparing it to someone. And we'll see, first of all, the variability in the healthy microbiome. And after we take a look at the healthy microbiome, we'll also discuss how this applies to medical applications and to understanding of.
I got a question. Why does every time I
Ilya, I'm so sorry to interrupt, but we can't hear you. I think you're on mute.
Sorry about that. Yeah, thank you. So, so what I was saying is that there is this in utero colonization hypothesis. This hypothesis is that there is a microbiome that is passed to the baby in the uterus. So Nick, I think there's somebody here that is not supposed to be here. Okay, so this hypothesis claims that there is a way to pass the microbiome to the baby from the mother in the womb. There isn't maybe enough evidence that that is conclusive. As a result, we won't focus on that hypothesis. But let's talk about some of the additional factors that do play a role in the development of the microbiome that have been studied extensively in infants, infants that are between 0 and 3 years. So, Nalika, would you be able to just remove the people
Yes.
that are?
Yes, Ilya, I'm doing that.
All right, so some of those include gestational age, so how long baby stays in the womb, the birth mode, for example, cesarean or natural, sanitation, antibiotic exposure is a major one, as well as host genetics. So several different studies have focused on trying to understand these factors in detail. And now we have some evidence of how the composition changes and how specific presence or absence of bacteria could be linked to some of these factors. Now, what happens in the early ages is that because this is an anaerobic condition, specific types of bacteria start developing. And what you can do, as I mentioned, the link that I just shared with you to the Silva database will allow you to search for some of these bacteria. So for example, we can take a look at Clostridia and search for this name in that database. So let's take a look at that. So you would go right here and just type in Clostridia and press on search. Okay, so as you can see, Clostridia, again, you can go kind of from the higher level of taxonomy all the way down to the more detailed one. So this is a firmicutes, right? Bacilli, erysipellatricalis, right? I'm not going to try and pronounce this, but you can see the variation right here at this level of how many different ones you're going to be able to find. And so you can zoom in. Okay, you can zoom in and you can see that there's a lot of variability. So again, there's going to be a lot of variability, and that is essentially what makes it so difficult to study this kind of variation at the lower levels, right? So the lower the level, the more detail we get, but at the same time, we're going to be getting a lot of information potentially from that. So here is an example of a study that was done on the effect of probiotics on the infant development, specifically infants that are preterm infants, preterm birth. So here you can see composition of the gut microbiome at the early phase of development dominated by 4 phyla: actinobacteria, Bacteroidetes, firmicutes, and proteobacteria, right? So we just saw that these are specific types of bacteria, right? And here we can see that it is dominated by these specific groups of bacteria. The adult microbiome, so again, this develops over 3 first years typically. So there's less of a diversity at the very early stage, and then that diversity grows and starts to become established during the adolescence. So from that early childhood all the way to the end of the adolescent period, the microbiome becomes established, and the primary factors that affect the established microbiome are the use of antibiotics, so how frequently and how intensively those antibiotics have been used, as well as the level of exercise and, of course, diet. And so here you can see the adult microbiome, and this is well characterized in the Human Microbiome Project, is really divided into these several different categories by the major factors that continue to affect the microbiome over the course of the adult stable condition. And those are specific enterotypes, 2 or 3 enterotypes that are well studied, body mass index, exercise frequency, different types of lifestyles, and then a major component are cultural and dietary habits. So here we can see a major thing that you want to think about as you kind of look at this healthy microbiome is diversity and balance, as well as this dysbiosis, so how it changes, how it becomes dominated by a particular type of bacteria that is maybe able to colonize and suppress other types of bacteria, reducing the diversity. And as a result, the function also changes. And so we'll see how that kind of looks like in the data itself. So first of all, let's briefly talk about these enterotypes. So different types of publications have been focused on understanding these enterotypes, but basically we can look at 2 or 3 different enterotypes. So here's an example. in specific types of bacteria, bacteriodes, Prevotella, and Ruminococcus, for example. You can see that they change in terms of their relative abundance. But basically, specifically those that are interested in the diet, they've been able to categorize these three different enterotypes into those that consume carbohydrates, like fruits and vegetables, then what we call the Western diet, so bacteriodes dominant types. And those are the Western diet because they're associated with kind of, the typical Western diet like meats, potatoes, and things like that. And then there's an additional cluster that is not as dominant, but it's primarily for those that are coming from rural agricultural communities. They don't have a lot of variety in their source of nutrition, and those are typically farming communities. So those are the kind of major emerging trends, right, that have come out of these studies. They've shown that there's definitely an association of microbial diversity because of the type of a civilization, so to say, or a community that the samples come from. So especially talking about the human gut microbiome, it could be categorized as very high in this remote hunter-gatherer population, kind of reduced, but still pretty high microbial diversity in the traditional farming or fishing populations, and then lower diversity in this Western European and industrialized populations. And so here you can see also how some of these variations have been associated with geography. So one major reason is because the same trend of industrialization could be tracked across geography, but also, not surprisingly, some of those habits associated with the type of food could be found within a specific population that maintains a specific aspect of lifestyle and food consumption. So a good review right here. Geography, ethnicity, or substance-specific variation in human microbiome composition and diversity. You can see, sorry, let me go back and share the link from this publication. I found it interesting if anybody's trying to understand more how this healthy human microbiome varies and what kinds of factors are there. Okay, now because of this variation, so again, think of the microbiome as a community. Once that community is stable, it assumes certain functions that are linked to immune responses to different infections. They form a film. A lot of times that film is protective. And so because of that, they're able to kind of help protect us from a variety of different infections, also by maintaining a certain immune response readiness for a variety of intruders that come. But because we also spoke not just about the bacterial communities, but we also spoke about the viral communities, these microbes are obviously involved in a variety of different diseases. And so the understanding of not only the bacterial diversity that we just spoke about, but also the potential of these viruses to be present and develop and get into the organs are very important. And here are some examples of viruses that cause human disease. So also something that we can look at as we study the microbiome. Epstein-Barr virus, the herpes family viruses in general, human papillomavirus, and hepatitis B and C are major conditions that drive about a quarter of cancers, right? So the link between microorganisms and human health and disease is very direct. And so we can see one perspective when we look at the microbial diversity and community that is in a in this holobiont relationship with the host. So it continues to coexist in a beneficial way with the host. And then we can see some of these becoming dangerous and causing disease. Okay, so now let's take a look at a couple of practical examples. what we will actually do during this training program. So in the training program, we will less speak about how this has been studied and less focused on literature review, but we'll more focus on the practical skills, how to actually take the data that you can find in some of these repositories and start asking questions. And what are the methods that you need to know how to use to be able to extract meaningful signal from the data? So the first example that we will use is the American Gut Project. So this American Gut Project is essentially a platform for citizen science microbiome research. And so a lot of the ways that we can start looking at this data is representative of semi-healthy, right? So either completely healthy or kind of, almost healthy, maybe some sensitivities and some preclinical conditions, but mostly healthy. So this was a project that started a few years ago, and essentially people were able to contribute their samples, answer a survey, a very detailed survey, as we will see, and as a result, you could compare your microbiome with other microbiomes that have been a part of this project. And also, as a result, researchers could start finding associations between certain types of conditions, and some of them, like we discussed. So one of the results that you got as a user, and this is one of the exercises that we will do in this program, is to try and replicate this kind of a report for an individual person, an individual sample, and compare it to some of the other samples that we have in the database. So here you can see an example of such a report. For example, what is in your American gut sample? You can see here how the proportion of these different types of bacteria is in your sample compared to a variety of other insights from the project. For example, average, similar diet, same gender, similar BMI, similar age, and this is your sample compared to some specific person. Your most abundant microbes, your most enriched microbes, and then you can see here some of these methods of comparison to other samples or collections of samples. So for example, we can see a major difference between different body sites, obviously. We can also see different ages and populations. And also you can see within one population, the difference, and this is the American gut population. So you can essentially, where are you in this population? So I think this actually is an older link that now has changed, but you will be able, what we will do in the program is we will actually take a look at the data that is available about all of these different factors. So here you can see one of the comparisons that we will try to do is use 16S metagenomic sequencing, Amplicon sequencing of these hypervariable regions V3 and V4, and try to understand whether we can find some variation based on regular or rare exercise, right? So we'll actually download the metadata. You will see that the metadata is quite complex. It comes with many different variables. A lot of the variables don't have data that we can use. So in all of this messy data, we'll try to select the ones that we can design a contrast for, and then we will select those, download the abundance of those OTUs, map those OTUs to the reference database, and try to use that information to ask the question, is there a specific difference between people who exercise never or rarely, or regularly and even often? And so one of the things that you will find is that this gives you a very good sense of the variability, right? When we see these results, these publications that find a link between a certain factor in the microbiome, we will actually get to experience that firsthand and see how that process is done. Another exercise that we will do is a study of the skin microbiome, which is an important factor in such conditions like acne. And one of the data sets that we will take is a comparison between perioral dermatitis, adult acne, and healthy skin. So perioral dermatitis is just a rash, like the redness of the skin. And then acne, of course, is a much more intense condition. And so one of the questions would be, is there a microbiome component to that? So obviously there might be other factors, but how does the skin microbiome actually affect those conditions? So here you can see a review article. So we'll go through this article as a review. and then we will take some data and we will be able to compare some of those samples to try to understand where is that variability and what defines each one of these conditions. And we'll also think about strategies to collect data that allow us to make get conclusive insights from such studies. Another interesting study that we will take a look at is the more indirect link between behavior and the gut microbiome. So the gut-brain axis is a whole topic of study for many different research studies. So what we will do is we'll take one of these example studies that talks about obese-type gut microbiota that induce neurobehavioral changes in the absence of obesity, right? So one direct association that we know of is the gut microbiome and obesity, right? And we can say, well, obesity itself causes specific changes in behavior. But in this study, they try to separate obesity from the microbiome and see whether the composition of the microbiome actually does have an effect on behavior. And so what they did is they took mice. Using antibiotics, they reduced the bacterial load, and then some were recipients of this high-fat diet or regular diet microbiome. And then what they did is they tried to assess at the end of this experiment whether there was an association, first of all, if that microbial community got established, and if it did establish, did it lead to some observable behavioral changes? And so they actually did some behavioral tests to see level of anxiety and things like that in the behavior. So after reading this paper and understanding the data, what we will do is we will apply a technique called a pipeline called Data2 that allows us to denoise the reads so that we have clear and clean operational taxonomic units, map those onto the taxonomy available in the Silva database, and from there develop a certain sense of how to process and visualize such data. First of all, using the Bioinfo platform, just to kind of see the logic of the analysis and the input, the output from each pipeline, and then turning to R to investigate further how we can modify and visualize the data to make our own interpretation of what we see in the results. So those are some examples. Beyond that, we will take a look at some of the other applications of microbiome sequencing and microbiome studies that are linked to precision medicine. And as you can already imagine from what we covered so far, because of the factors that we've observed with obesity, with the type of diet, with the exercise levels, antibiotics use, etc., there's obviously a lot of those things could be influenced during the life of a person. And so as a result, the conditions that we will see for example, related to disease, are also affected by some of these things. So these could include dietary interventions, prebiotics, probiotics, and even transplantation of the microbiome to see its effect on specific treatments or development of specific conditions. So we will take a look at a number of studies, including studies that have led to translational research results. For example, here, we can see China approves Alzheimer's treatment that targets the microbiome, right? So we will take a look at some of those potential interventions that are being explored now for different types of diseases, and in general, as a component of precision medicine, right? So not only how do we focus on a specific type of a treatment that could influence the microbiome composition or microbiome functionality, but how can we also incorporate microbial community understanding into an approach for precision medicine? How can this be used for cancer therapies or therapies in general, including kind of prebiotics before the treatment or during the treatment or after the treatment, right? So we will take a look at some of those studies as well. So to summarize, the program will cover in several different sessions an introduction to different types of sequencing techniques, different data repositories. We will take a look at some curated examples that we have selected to illustrate some of the approaches for gut, skin, microbiome studies that allow us to understand differences between healthy variation in healthy representatives of microbiome, like the Human Microbiome Project, or associated with disease. And we will also take a look at ways you can find your own data sets on repositories like NCBI that can help you ask questions about different types of diseases. Someone mentioned cancer. I mentioned Alzheimer's disease. Obviously, there's a lot of types of diseases associated with the gut. And we will also take a look at other other applications that you can easily find on NCBI. Now, the goal of this program is really to prepare you to be able to independently go from identifying a data set and then using metagenomics to answer a research question. And one of the ways that you can get started, and this is relevant for anyone, whether you will be joining this training program or not, we have a free resource on the Learn portal that you already got introduced to in the beginning of today's session, where you can take an introduction to metagenomics. So let me briefly point you to that free course. If you already created an account, you will be able to complete this whole Bytes and Molecules course for free. And one of the lessons in this course is an introduction to metagenomics. So again, let's go to the Learn portal, and I will show you how to find that. So if you are here on the Learn portal, you'll be able to go to the Courses tab. And right here, there's a search bar. So you can simply search here for Bytes and Molecules. So this is Course 2, Bytes and Molecules. And I will post the link to this course in our chat as well. Okay, so here, once you scroll down, you will see Introduction to Metagenomics, and some of the material that we covered in today's session is going to be available there as well with some links. So if you are not familiar with some of the terminology that we used in today's session, if some of these things sound interesting, but you don't feel confident that you will be able to go through all of the practical sessions, we highly recommend that you go through some of this material before you start. That will give you a sense of how the material is organized and what you will be able to get out of it. Obviously, this is a very introductory type of lesson, but it will give you a good sense of what we will do. Now, also inside of this introductory course, you will see some of the other things that we will talk about. So it's useful to at least take a look at all of the things that are present, including those of you that will be interested in doing more with your data. We will be using the T-Bioinfo platform, which I will demonstrate in just a second, but also we will be focusing on some data visualization and annotation techniques using R. So if you're not familiar with R at all, you don't know anything about coding, we highly recommend this Introduction to Bioinformatics Languages lesson, where you will be able to learn about the syntax, install and kind of make sure that everything works on your own system, and then also be able to try running the code first in the browser and then copy that code to your, once you're done, you will be able to open that in your own environment on your own system, like in RStudio, and try to run it here. So I'll just give you here an example. It's not only good to kind of be able to run the code, but also to understand the code, and you will be able to check these different things that are made here. Okay, so I will pause here, and I wanted to ask if there are any questions that I can answer right now. We're not going to allow audio questions at the moment, but if you want to put those questions in the chat, I will go through them one by one. Okay, how to access free classes, and can we provide you with further webinars? Yes, so after you complete this particular course, so again, this webinar is an informational webinar. We kind of talk about some of the things related to the topic, and we will introduce other webinars in the future. We actually have a student organized club called the Omics Club, which will include free sessions with guest speakers, and you're welcome to join those. But if you want a certificate, all you need to do is you need to create a free account on this portal, and then go through the Bytes and Molecules course. Here under your profile, once you complete that course, again, here you will see, for example, these are some of the courses that I'm working on. And once you complete a course, you will be able to get a certificate. So in fact, if you go here to the certificates tab, you will find here all of the certificates that are related to the coursework that you completed, as well as to the specific programs if you are interested to join. Okay, so I see another question. Which part of 16S is best for sequencing for pair end? Which length is the best? So these are great questions. So again, we'll go into the details, but just for everyone else that is here and would like to know a little bit more about 16S sequencing itself, I will just show a slide, just one moment. Okay, so typically, you can see right here for 16S Amplicon Metagenomic Sequencing, this is the full 16S ribosomal RNA gene. Now, this gene is conserved across bacteria because the ribosome obviously has an important function for all bacteria, but there are hypervariable and more conserved regions. So these regions right here are more or less conserved, and then here you have variable regions. So typically 16S amplicon sequencing combines V3 and V4 hypervariable regions. These regions together fit nicely into the typical length of the 16S or of the systems like MiSeq. So MiSeq is useful because the sequencing length of those matches, about 300 nucleotides long, so it matches the sequence of those hypervariable regions. Okay, another question. Is there an option for a research publication? So this program that we're discussing today is a training program. So this will introduce you to some of the skills that you need to know, make you familiar with some of the data sets that you can use for your own analysis. If you end up developing an analysis, we highly recommend that you go for the advanced level. The advanced level is designed to allow you to have a little bit more time and more interaction with our mentors so that you can start an independent project. So just to kind of explain what this independent project is, right here, if you click under your profile to projects, you will be able to start a project after you complete the training. So after you reach 10,000 points and you're ready, your profile is ready, you've reached 10,000 points, you will be able to start a project. These are the projects that others have completed in some of the training programs. Now, a lot of times these projects take quite a number of months to complete. And that's why we offer a special program called the Research Fellowship that you can apply for. And if you're accepted, then you can work on a research project with one of our mentors with support and resources that you would need to be able to complete that project on time. So also in the chat, you will see that Subrajit Barua, who is in charge of our scientific communication, is posting information about the Omics Club. There are 2 exciting free webinars coming up on metagenomics on January 8th with Gareth Trubel, who will be talking about unraveling the hidden potential of active microbial and viral communities. And also on January 21st with Dr. Christopher Taylor, who will speak about metagenomics and microbial communities in general. So everybody's invited to join. The details are in the chat, and how you can join is in the chat as well. So if you're interested to kind of learn more about the applications of metagenomics and microbiome studies, you're welcome to join those. And if you need any more information, please make sure to reach out to Subrajit Barua. Subrajit, maybe you can post your email address there, so if people have any questions, they can reach out to you. Okay, so thank you again for letting me introduce the subject today. I will now pass it back on to Sonalika to speak about registration.
Thank you so much, Ilya. Okay, so this is the pre-registration page where you all had pre-registered yourself for the program Metagenomics, and you will be able to find all the details of the syllabus and also the outcomes of each and every session, the topics and the outcomes and the associated resources as well on this page itself. And while you scroll down on the page itself, you will find 4 live sessions that we have planned for the session. And these 4 live sessions will be on overview of methods and applications for metagenomic sequencing and analysis, big data analysis platform, metagenomics analysis on the Bioinfo server. So here we'll be training and also get some good hands-on practice with the pipelines on the server. And to mention, the participants of the metagenomics program will be getting the educational license where they can run their pipelines on the server and analyze the data as well. The 3rd live session will be on interpretation and meaning, that is statistical analysis, visualization and interpretation using R. And the 4th one will be a very interesting one, which includes the future directions for full metagenomic sequencing, metatranscriptomics, and functional analysis. And we'll be having 2 guest speaker sessions as well, where on January 8th, that is actually before the program begins, Dr. Gareth Trouble will be talking about unraveling the hidden potential of active microbial and viral communities. And the 2nd guest speaker session will be by Dr. Christopher M. Taylor. And we have Dr. Chris here with us. So we welcome you, Dr. Chris. And the session will be on metagenomics and microbial communities. And this session will be on 21st of January at 3 p.m. CST. So here is the whole schedule of the program, and also now let's talk about the registration. So to register yourself for the program, so here we received your pre-registration as your interest to invite you for the free webinar. So many of you get confused with both of the registrations type. Now to enroll yourself for the program, you need to visit the program page on the learn.omicslogic portal. And you will be able to find the link here in the chat box. Okay. So once you are here on the program page, again, you'll be able to first of all see the whole session schedule along with the dates, the topics to be covered and the associated resources. And these all resources are linked. So this is for the program participants that once the session has been completed, you will be able to find, I think the session dates are to be updated. Okay, so once the session has been completed, you will be able to look at the session video under each of the sessions. That is, after each session is completed, the recording of the same session will be uploaded under the sessions tab. And also you can watch and go through the recording and complete the resources here on the program page itself. Now to enroll yourself for the program, you need to scroll down where you will get 3 levels. The first one is the beginner level, the second is the intermediate level, and the third one is the advanced level. Now, what is the difference between these 3 different levels? So in the beginner level, you all will be able to be a part of the group sessions that we are going to conduct. And the intermediate level, those who require some extra training or have some extra questions to ask or need more precise help from the mentors, then they can go ahead with the intermediate training where they'll get one-on-one mentor support. And in the advanced license, the program participants will be able to work on a research project, and for that research project they're going to get some extra time period along with the usual 1.5 month time period of the program. So that is the difference between these 3 different level types. Now you can enroll or like pick any of the subscription levels that answers your requirements the best. And also when you scroll down, you'll be able to find a list of the program coursework that is associated with this particular program and the certificate that you will be getting on the completion of this particular program. So that was the simplest way how you can enroll yourself for this particular metagenomics program. Now, if you have any queries, please let me know in the chat box so that we can answer your questions. Okay, so if there are no more questions, I would like to put in, oh, we have a question. Yes, thank you, Sparsh. So you can reach out to us at marketing@omicslogic.com for any of the queries, and we'll be able to help you out with your questions and assist you with your enrollment for the program. So the first session of the program is on January 10th, and the first session is a free session, and everyone can be a part of the session and go through the introductory topics at first and get insights of what all would be covered and how the program would be working. So that was all for today's session. I would like to thank you all for being a part of the session. And if your questions were unanswered, or if you have any other question that might struck after this particular webinar, please feel free to mail us. Yes, definitely, Alex, there are career opportunities. Now, all these training programs that are being conducted, they are designed in this way. So as Ilya also mentioned, we focus on project-based learning. So yes, there are definitely career opportunities that you will get, like you will be able to get yourself a lot of these opportunities after completing these sorts of trainings that Omics Logic conducts, which will help you sharpen the topics definitely, and also help you give a clear picture of how the concepts or the methodologies could be applied to solve a real life research problem and do the analysis. Yes, Satya. Satya, we will, for your questions, Sparsh will be reaching out to you, and she'll let you know like what the research fellowship program is and how it works and how you can proceed from a research fellow to an intern with Omics Logic. And please put your mail ID in the chat box so that Sparsh can reach out to you, or you can directly reach out to marketing@omicslogic.com. Yes, Sophia, we have few scholarships for the program as well. So we'll be able to share with you all the scholarship form. So just after this particular webinar, I'm going to share the recording of the webinar. And along with the recording, I'll also share the scholarship forms that the participants can fill out. Now, in the scholarship form, you are required to submit your curriculum vitae, and after reviewing your application, we will let you know what scholarship we can provide you for this particular program. Now, to make sure that you receive the mails from our end, please make sure that you add marketing@omicslogic.com to your mail address book, because we have been interacting with a lot of users. Sometimes the mail gets into spam or maybe promotional box. So to get the mails in your inbox, don't forget to add the marketing@omicslogic mail address to your address book. Any other question? Okay, thank you, Satya, for sharing with me your mail address. I'll be able to share this with Sparsh Thar, and she'll be reaching out to you with your question on a research fellowship program and how you can proceed further with the internship opportunity. Okay, thank you everyone for joining and being a part of this webinar. We'll now meet you all on January 10th and get started with Omics Logic Metagenomics. Thank you.